Welcome to solverfy.
Hello students, the question asked is why is the D antigen considered to be the major antigen of the Rh system? The D antigen is considered to be the major antigen of the Rh system because it is the most immunogenic and clinically significant. It is responsible for majority of the Rh-related transfusion reactions and hemolytic diseases of the fetus and newborn. Another question asked is an individual lacking all antigens of the Rh blood group system is classified as what? They are classified as Rh null. If they lack all antigens of the Rh blood group, they are classified as Rh null. Another question also is the theory of inheritance that claims that the Rh antigens are determined by three closely related sets of allelic genes was proposed by who? It was proposed by Fisher and Ray. Another question asked is, with few exceptions, why do blood recipients not require weak retesting of their red blood cells? Blood recipients do not require weak retesting of their red cells because weak D antigens are not clinically significant. And they do not cause transfusion reactions in fetuses or newborn baby. Another question asked is, why does the term antithetical mean with regard to the blood group antigens? Antithetical means that two antigens are mutually exclusive. Which means that the presence of one antigen implies the absence of other. An example of, for example, Rh factor blood system antithetical antigens is that D means the presence and D means the absence of the antigen. When D is present, D is absent. When D is absent, then this will be present. Another question asked is, if D negative red cells of genotype R are transfused into a D positive recipient of genotype R1, what antibody is most likely to develop? So the antibody most likely to develop is anti-D antibody. Another question is, in what ways can the formation of Rh antibodies be stimulated? The formation of Rh antibody can be stimulated by transfusion of Rh incompatible blood. Pregnancy with Rh incompatible fetus or injection of Rh positive blood products. So due to this, there will be Rh antibody. There will be a stimulation of Rh antibody. Another question is, if parents who are heterozygous for D antigen will have what likelihood of producing an Rh negative child? The parents who are heterozygous for D antigen will have 25% of likelihood of having an Rh negative child, because one of them will be a homozygous dominant, two of them will be heterozygous, and one of them will be Rh negative, which means that 25%. Another question also is, Rh antigen that is a combination of the C and E antigen. So we are supposed to tell which Rh antigen is combination of C and E antigens. Is the antigen, it is the Rh antigen which is called as G. Another question also is, the G antigen is present if what or what antigens are present? C and D. C and D. Another question asked is, what is the clinical significance of Rh null phenotype? It is significance is that individuals with this phenotype, which is Rh null, can develop antibodies against Rh antigen, which leads to the transfusion reactions. Another question asked is, a donor initially tested D negative using commercial anti-D reagent. The weak D test was positive. The RBC unit should be labeled as D positive or D weak positive. So the RBC unit should be labeled as D positive, both D positive and weak D positive. I hope you found the video useful. Thank you.